I think most of my previous speakers already covered everything, what I'm going to tell, so it will be easy. So just a quick view of the talk outline. I'll just talk about the history of our involvement in PBCRC, the collaborators, research themes, impacts, and a little bit about where we are moving from here. History, I heard from my older colleagues that, QDAF, earlier called DPI, started working on post-harvest grain protection research in 1950s, early 50s, the Bruce Champ days. So it's a very old group. It's got a very strong focus on biology and fumigation research and grain protection developments. So we played a major role in the CRC NPV, the first phase of PBCRC or the CRC work. And Pat already covered a lot of those projects. And from 2006 to 2012, mostly the projects revolved around the themes of resistance monitoring and management. We played a major role as well in PBCRC phase 2012 to 18, and approximately $3 million was invested in our lab in Queensland. The Program 3, most projects came under Program 3 is safeguarding trade, and it addressed the Commonwealth milestones like developing tools, technologies, and strategies to safeguard our grain markets and better management of established pests. Most of those projects actually built on the success of the previous projects during the CRC and PB time. And both phases, actually the major industry issue was the strong level of resistance developed in the rusty grain beetle, the Cryptolestis ferruginous. I must say that this is the strongest level of resistance in any store grain pest to phosphine recorded so far. So these are our collaborators. We were very fortunate to have collaboration with a wide range of research organizations and academics, including Kansas State University. Tom Phillips' lab was involved there. Our oldest collaborator in Paul Ebert's lab in UQ, not in terms of age, but in terms of period. Murdoch University, and we have several growers working with us, and the major grain bulk handling companies led by senior people. So these are the three major themes we run our projects. One is the National Resistance Monitoring Program. It used to be 3 fragments run across the country, like the New South Wales Department of Agriculture and the DAFWA in West, and the northern region was run by our lab. But the first time under PBCRC umbrella, we came together. We, we actually developed a national protocol to run this program, and so that we can compare our results properly. And I'll go a little bit later on, what we achieved through that. And ecology, Greg ran 2 successive projects there, and there was another project on the Cryptolestis lure being developed. And these are the groundbreaking research, as Pat mentioned before, happened in the ecology area. We never knew about what happens to insects outside the storage environment. Resistance management, we had 2 successive projects where we actually developed an integrated approach, utilizing all tools, maximize the potential of all treatments available for the benefit of the industry. Those last 3 projects I listed there, we played a role as a collaborator, and one with Paul Ebert, and 2 run with PBCRC leadership. So I'll go quickly to the impact. The overall impact, you can see these projects actually helped maintaining Australia's global reputation as a reliable supplier of insect-free, high-quality grain. That's the bottom line. So we actually established best practices for pest and resistance management for the industry here. And that helped in extending the usefulness of phosphine. I'm not going to the details because it was all covered. We actually developed sulfur fluoride, another fumigant, as an alternative. And we actually suggested industry should protect that fumigant as well, to use it strategically only as a resistance breaker, not to overkill it. So it's not only developing protocols, but giving that sort of advice. And as previous speakers already mentioned, new knowledge on ecology of key pest species, and like the flight across the storage landscape, the gene flow, and their relatedness. Resistance monitoring, we established a system where we can give early warning of new resistances, where they are actually developing and how far they are spreading. And that actually helps the industry for the timely containment and eradication of that particular resistance population. And we have a 30-year-old database called Australian Grain Insect Pest Resistance database. It's such a huge database. It helps us in analyzing that, utilizing some modeling and seeing the frequency and the spread of resistance across all over Australia over the years. And we published 4 papers already from that database mining. And that database also got information on the storage types, the commodity, the temperature regimes, all sort of data there. So that actually helps us also establish the causes of control failures and the factors responsible for selection of resistance. So that's another key element of that research. First time we developed a quick test. The traditional way of diagnosing resistance in a pest population is at least 14 days or something. We actually developed a test where we give industry advice on a population the same day within 5 hours of receival of the pest strain. That's been published. And we also established the resistance detection diagnostic dosages for 2 new chemicals, sulfur fluoride as the new fumigant and spinoside and new grain protectant. With ecology, just like a snippet, like the flight dispersal, away from storage. Everybody thought that, insects are inside the storage, how to kill them. Nobody thought, where they're coming from, where they're going, all sort of things. So that project also helped developing the sampling trapping methods best utilized for monitoring. In the management, we developed an integrated strategy utilizing all these new protocols and time to reinfestation. What happens, after a successful fumigation? How much time it takes for the pest to come back? And the last one I will touch on is the adoption of new knowledge. It's a practice change. We got an extension network. We got extension specialists in field model here. And all those information going to growers, going to end users through that network. And it also helps in updating the national resistance management strategy. Science impact, there are several publications came out from this set of projects, 14 research papers, 4 extension articles, 16 conference presenters with few keynotes. You can see the publication in science, heredity, genetics, pest management science. We got the annual review article. We got a new invitation to publish another annual review of entomology article on phosphine resistance. So who are benefiting? As everybody know by now, where to now? We know that we are containing the resistance to that 10% level. We have good collaborations in place, but we need to work with industry again to implement all the changes and the practice changes, and we got to go for future research with industry support again. Thank you.